Hello. Now we're going to look at Chapter 22, the digestive system. This gets to be a large amount of information, not because, and I could make so many puns about, digesting all of this information and chewing on this thought. And yes, I know. Oh, those are terrible puns. But there is a lot of content here, so I want to advise you now. It's important if you cover it all, pre-plan your studying, because this is extremely important. It is going to be not only important for allied health, but if you do medical microbiology, general systemic health, in other words, the health of the human being, in part depends on a properly functioning digestive system. Well, let's get going. First off, when we talk about the digestive system, it consists of the digestive tract and the accessory organs. Now, digestive systems consist of, when we talk about the digestive tract, which is also referred to as either the gastrointestinal tract or the alimentary tract, you have a variety of structures: oral cavity, pharynx, esophagus, stomach, small intestine, large intestine. That's the basics. But then you have a variety of accessory organs that assist in the mechanical processing of food and the chemical processing of food, usually by providing secretions that allow for the chemical breakup of the food to its more basic components. Example, proteins broken down to their subsequent amino acids. Now, the structures that are considered accessory organs include teeth, tongue, salivary glands, liver, gallbladder, and pancreas. When you think about it, excuse me, what the digestive system does is it takes complex chemical components, okay? Whether it's the Twinkie you had, the steak you had, the salad you had, whatever, breaks down what you would use for energy. Starches are broken down to eventually monosaccharides. Lipids are broken down to fatty acids, monoglycerides. Proteins are broken down to the amino acids. Then we have them transferred and transported throughout the body by the cardiovascular system, which, by the way, has to also go through a round of external and then eventually internal respiration, because, of course, blood has to be oxygenated, tissues have to be oxygenated. Eventually, any of the metabolic wastes that are used or produced, I shouldn't say used, but are produced as a result of metabolic activity will eventually be disposed of as waste in the urinary system. So if you look at this from respiratory system, systemic tissues, digestive system, cardiovascular system, and urinary system, one of the big four is the digestive system. And as you see in this diagram, the major organs of the digestive tract, you have the oral cavity, the pharynx, the esophagus, the muscular tube that delivers the food, the bolus of food down to the stomach. Of course, the stomach, and then we start with the small intestine, which is composed of the duodenum, the jejunum, and the ileum. Pretty much a lot of the material have been absorbed. A lot of the essential nutrients have been absorbed by the time you get to the large intestine. There is a little bit more that is absorbed in here, the large intestine, but also you have the undigestible material, as well as, let's see, 1/3 of the fecal matter is consisting of bacteria cells, interestingly enough. And all of this waste is processed and passed through the digestive system down to the sigmoidal colon and eventually passes out of our body by the anus. The accessory organs would be, of course, the teeth, which are not shown in this diagram, which are doing the physical or the mechanical breakdown, grinding up, cutting up, chewing up of food. The tongue, which mixes around this food and mixes it also with salivary gland secretions. We also have the liver, which also secretes its own material. One, it secretes bile, which is an emulsifying agent for the proper breakdown of lipids. Two, we have this gallbladder here, which acts as sort of a temporary storage for extra bile. And then we also have the pancreas, which provides bicarbonate ion solution. The bicarbonates are necessary because when you add acid from the stomach, you have to neutralize this, otherwise it would cause profound damage in the small intestinal lining. It also provides a variety of enzymes which are necessary for the breakdown of proteins, lipids, and carbohydrates. Now, when we talk about the digestive tract, it's a muscular tube lined with mucous epithelium. It's very long tube lined with this mucous membrane. And when we talk about the mesentery, this is double sheets of peritoneal membrane. These are the sites of blood vessels, lymphatic vessels, nerves that will eventually interact and penetrate into and support the digestive system organs. You see, what the mesentery do is they stabilize and attach the organs in the abdominal pelvic cavity. Keep in mind, cavity, empty space. So if this stuff is hanging around in there, it can't just jostle from one side to the other. Furthermore, it prevents intestinal entanglement during digestion or swift body motions. What you don't want is having your intestine tied up or kinked in such a way that blood flow is not allowed through the intestinal area. Now, the layers of the digestive tract will always come up as mucosa, submucosa, muscular layer, and serosa, which is the outermost layer. When we talk about secretory glands in the mucosa and submucosa, they're carried to the lumen by the ducts. So the secretions come up to the lumen. You will have also very noticeably circular folds, particularly in the small intestine. These are referred to as plicae circularis. They enhance the surface area. They're permanent folds in the lining. Then what you have is the muscular layers, which consist of smooth muscle. You'll have a circular layer and a longitudinal layer. When the longitudinal layer contracts, it's as if you're shortening the length of the intestinal segment. The circular one, when it contracts, it'll be as if you're pinching off or having the intestine decrease in its lumen space. Now, there's an advantage here. This allows you to do what is called peristalsis, where you have a pinching off and then you have this running forward in a coordinated fashion of this pinch, so it pushes along food or the chyme, the food-like material, through the intestinal tract. Now, in the components of the mucosa, you're going to find villi. These are extensions, they're very fine cells that will enhance absorption. Underneath them, the lamina propria, and underneath that, the muscularis mucosa. When you get to the submucosa, you're going to find that there's going to be a neural plexus. This is a neural network superficial to the muscular layer. Now, this contains sensory, sympathetic, and parasympathetic fibers. This is absolutely essential for the coordination of movement, but also of sensations of, full, hot, cold. Also, if blood flow has to increase or decrease because of either sympathetic or parasympathetic stimulation. Remember, in parasympathetic stimulation to help you, blood flow is going to be increased to meet the absorptive needs of the digestive system. Parasympathetic is basically been considered rest and digest, whereas sympathetic, you're not going to really care about digesting a meal. It's, basically fight or flight. So the blood gets reduced in its flow to the digestive system so that the extra blood can be used for providing further service to the skeletal muscles to enhance that fight or flight capabilities. Now, the myenteric plexus is actually between the circular and the longitudinal muscle layers. Contains neurons of the enteric nervous system. which controls most of the digestive activities, as well as the sympathetic and parasympathetic influences as well. And as you can see here we have the mesentery. You can see the movement and penetration and distribution of the mesenteric artery and vein. You have the mesothelium. You have the areolar tissue, which is thin, smooth, loose connective tissue. You have the mesothelium on the other side, so you've got two layers. Then what we start dealing with is the major layers of the digestive tract, so that'll include, we have, of course, the lumen here, and right first we're going to have the mucosa. What do you see here? These circular folds. These circular folds enhance the surface area. The next level of enhancement would be, of course, where the villi are here, but we'll get more into that later. Then underneath this, you have the submucosa. And beneath the submucosa, you've got your muscular layers, both circular and longitudinal. And then the outermost layer, which is kind of this connective tissue layer, called the serosa. And if you notice that the serosa pretty much is in contact with the mesothelium. Let's go down further here. And I've talked about the circular, the plicae circularis, circular folds. You see here secretory glands in this structure. You see the villi, okay? And now on the tips of the villi, they're going to have actually extensions of microvilli. This is all referred to as the brush border, because what you're really doing is enhancing further and further absorptive surface area, which increases the capability of getting all the nutrients out of the food that you consume. Now you'll also notice here submucosal glands, as well as intestinal glands, which are going to secrete mucus, and it may be modified, may be somewhat more alkaline. It may contain such capabilities to allow lubrication as the material is passing down through the lumen of the intestinal lining here. You will also see lymphatic vessels in the submucosal layer, and of course arteries and veins. Notice that the circular layer is on the inside. Just inside of the circular layer is that submucosal plexus, and between circular and the longitudinal muscle layer you have the myenteric plexus. So you have several different layers of neural networks within this hollow organ to coordinate the proper function, the flow, to respond and provide certain information if there's evidence of nutrients, if there's evidence of pathogens, if the stuff comes in super hot, super cold, all that type of stuff, okay? Now, as I mentioned earlier, the smooth muscle tissue is found throughout the body. But here it's going to play a particularly prominent role in the digestive system because smooth muscles are long and slender. The circular layer will reduce the lumen, all right, and the longitudinal layer will contract the length of the tube. The structure of the smooth muscle is thin filaments attached together by dense bodies, and adjacent smooth muscle cells are bound together by these dense bodies. Upon contraction, the muscle cell twists like a corkscrew. So in other words, what you want to do is get a lot more traction as you have this contraction occurring. As you can see in this layer here, in this micrograph, you have the circular layer. And then what you see down here is the longitudinal layer. You notice how the direction of the nuclei, etc., it's almost as if it's more lined up and stacked on top of each other for the cells. These are necessary to help push along the products so nothing stays at a particular site too long. Here, and this is something that we've probably seen earlier in AP1, are an example of the arrangement for the smooth muscle cells. When they're relaxed, you can see a sectional view. You see where the dense bodies are. Notice that you have thin and thick filaments. The filaments are, of course, attached to the dense bodies. When you have it relaxed, you have this meshwork that you can see of intermediate filaments attached to the dense bodies. And when you have it contracted, you can see how this is beginning to be kind of scrunched up. But if you could have a three-dimensional view of this, it would be almost like a spiral approach or spiral structure. When we talk about the different types of smooth muscle, the multi-unit smooth muscle cells require stimulation by local motor neurons, or more than one of them. The visceral smooth muscle, which is due to pace setter cells and gap junctions, the smooth muscle contract via nearby motor neurons, but they act together in concert during the contraction. Then we have pace setter cells. These are cells that cause smooth muscle layer to contract on its own, spontaneously, at regular intervals, basically to maintain the muscle tone. Now, with multi-unit smooth muscles, you can see how this is kind of, you have the neural network sort of in between, sandwiched in and out as a meshwork in between all of the smooth muscles. Okay, that's nice. Visceral, think of visceral like the viscera of the small and large intestine, etc. Very different. Neurotransmitters on the top. It basically releases via the varicosities, those are the round structures again, and it rains down on top of the first layer. But because you have these various gap junctions, the signal gets passed down to subsequent layers of the cells, and you're going to have basically stimulation across the cell-to-cell connections. And you will also have the presence of pacemaker cells, which on themselves will every once in a while cause slight contractions to keep up muscle tone. Okay? So in this situation with the digestive system, you're going to see much more of it being visceral smooth muscle. Now, smooth muscle contractions produce motility of the digestive tract. I've mentioned that before. There's actually several things to keep in mind. One is, of course, peristalsis. This is the wave of contraction in the circular muscles that forces the bolus of food forward in the tube. For example, when you swallow and you've swallowed a compact mass of food, it goes into the esophagus and you start having this coordination of contraction of the circular muscles, which will force the bolus of food forward in the tube until you reach the stomach. There, a sphincter will open up and the completion of the peristalsis, which will basically force the food into the lumen of the stomach. Segmentation is a little bit different. Now think about this. Peristalsis, you're moving forward. Segmentation is kind of a mixing up situation. Segmentation is observed in small and large intestine. These are coordinated muscle contractions that result in the churning or fragmentation of the bolus. It's kind of like leading into mixing of the intestinal secretions as well. So you have this up and down, up and down, up and down, up and down throughout the whole length of the tube, with the idea of being able to churn and mix and turn over all of the food material so that it's adequately saturated with the necessary, whether it's enzymes or neutralization by bicarbonate ions, etc. Here we have an example of peristalsis. You can see the bolus of food arriving in the digestive tract. So basically you're heading in a particular direction from mouth toward the anus. What you have are the coordinated contractions by the circular muscles. Then you have this coordination by the longitudinal, which will pull forward. And so you have this contraction occurring in fits and starts of the circular muscle as you have further assistance by the longitudinal muscle, and you're pushing along this particular bolus of food. In segmentation, a little bit different. Everybody gets to contribute in this coordinated contraction and relaxation. Contraction, relaxation, contraction, relaxation, etc. The idea is breaking it into smaller lumps, mixing it sufficiently, turning it around and breaking it again into smaller lumps, etc. And you have to keep in mind that there are local factors that interact with the neural and hormonal mechanisms to regulate the digestive activities. So no one system plays complete priority over all the other systems. It is a group effort, if you want to think about it that way. Local factors are those triggers such as changes in pH, physical distortion of the digestive tract, presence of selective chemicals. Now, the neural control mechanisms, this is when you have the myenteric reflexes, the long and short, play a role in the segmentation of peristalsis. Short reflexes is due to chemoreceptors or stretch receptors in the walls of the digestive tract. Long reflexes include parasympathetic motor fibers to control movements. The central nervous system plays a role in secretion and digestive motility. Also, we have hormonal mechanisms. Now, there are at least 18 hormones that play a role in digestive function, including hormones produced by the enteroendocrine cells. We will get into some of those later. Yes, you need to know them. So you can see this multiple step process where you have local factors. One, do we have a stretch? Do we have changes in pH, etc.? Then we start having further stretch receptors, and we have the long and the short reflexes that are involved with the CNS, which will lead to peristalsis and segmentation movements. Finally, we have the hormonal control mechanisms. Some of this is played in a role from the enteroendocrine cells as well as secretory cells. There are hormones that are released. The bloodstream will transport them back to certain secretory cells to cause the release of buffers, acids, enzymes, etc. The digestive tract begins with the mouth and ends with the anus. That we've talked about. So we're going to start going through some of the structure of these parts of the tube first. Then we'll get to accessory glands later. The mouth begins the chemical processing and mechanical digestion. We have the pharynx, which is involved with transport, muscular propulsion of materials to the esophagus. The esophagus is transporting materials to the stomach. The stomach, there you have further chemical breakdown by acid and enzymes. The mechanical breakdown is by muscular contractions. The small intestine, you have further enzymatic digestion. You will start to begin absorption of water, organic substrates, vitamins, and ions. And also you will eventually lead to the remnants coming in contact with the large intestine. Now, this is where they say dehydration, the removal of water. To help you understand this right off the bat, a lot of water in the form of secretions has been added to this food, and it has to be recovered. And that includes secretions from the salivary glands, from the stomach, the pancreas, and all the way throughout the entire small and large intestine to a degree, much more the small intestine, where you have some liquids that are necessary to keep lubrication and keep materials moving, okay? But by the time you hit the large intestine, first thing that's going to happen is that the fecal remains are going to be having a lot of removal of water. You will have them being compacted into of the indigestible materials, and that includes, as I mentioned before, bacteria cells. And then you will have the preparation for elimination. And here we can see the major organs of the digestive tract here, accessory organs again. So let's talk about the functions of the digestive tract: ingestion, mechanical digestion, propulsion, chemical digestion, secretion, absorption, defecation. By the way, what do I mean by secretion there? You have to secrete a large number of certain materials, particularly enzymes, to help break down these complex chemicals that are in food. Break down your Twinkie. All the sugary goodness of it has to be broken down, because in a lot of cases you have sugars that are more disaccharides. They're going to be broken down to monosaccharides. Any starchy material that's present You have to have that broken down from being a polysaccharide to a monosaccharide as well. Okay, that's just one example. Okay, and then you have to absorb it. And some things absorb better and get transported better in the blood than they do in the lymph nodes, or actually they get absorbed not very well in blood immediately, but get transported easily and better in the lymph system, particularly lipids, but we'll get more into that in a bit. And then we have the expulsion of the remnant waste and defecation. Now, the oral cavity is a space that contains tongue, teeth, and gums. The oral cavity is lined by oral mucosa, which is a stratified squamous epithelium. In the sagittal section, you have, of course, the hard palate and the soft palate. The anterior and lateral boundary of the oral cavity, you have the labia, which is the lips, the cheeks, and the body of the tongue. The inferior boundary of the oral cavity is the floor of the mouth, which is inferior to the tongue, supported by the glenohyoid and the myohyaloid muscles. The posterior boundary, it consists of basically the uvula, which prevents food from entering the nasopharynx, the palatine tonsil, the root of the tongue, and the lingual tonsils. As you can see in here you have sort of a bony structure, that's our hard palate, and this is more softer tissue, which is the soft palate. Here would be the uvula. This would fold back as we swallow a bolus of food. Notice that you have several tonsils here that are clearly in view, the palatine, the lingual, and don't forget way back up here is the pharyngeal. We have, of course, the tongue. This is the body of the tongue here. The floor of the oral cavity consists of muscles of the glenohyoid and the myohyaloid muscle. Okay, and let's move on. The anterior view includes, and this is again of the muscle, the oral vestibule, the frenulum of the upper lip, the frenulum of the tongue, and the lower lip. The palatal arches, the palatal glossal arch, extends between the soft palate and the base of the tongue. The palatopharyngeal arch extends from the soft palate to the pharyngeal wall. Fauces are the space between the oral cavity and the oral pharynx. And then, of course, the tongue and the gingiva. By the way, the frenulum. Let me see if I can bring up the image very quickly. The frenulum is this little attachment right here. This is the lingua frenulum, which is the frenulum of the tongue. Here is the frenulum of the upper lip. Now, just a sidebar, if you ever seen those murder mysteries and they go up and they say to the medical examiner, well, doc, how was this person found in bed and no evidence of trauma and they died? And the doc looks and says, oh, no, they were murdered. And they're like, how do you know? They said, well, they had a pillow put over their face. They went, pillow? We don't see any feathers anywhere, and so on and so forth. A natural response, if one is being smothered, is for them to violently move their head side to side, just as if you're saying no. Now, if you're putting pressure with somebody over that with a pillow, what will the ME will be able to tell is that the frenulum of the lower lip and the upper lip, it's usually torn away. Okay? Now, you also want to be mindful of the fact that we said gums in here. These are the structures around the teeth. We'll get much more into the individual teeth in a bit. Okay? Teeth in different regions of the jaws vary in size, shape, and function. The tooth consists of an enamel covering, which is a calcium phosphate crystalline matrix, dentin, which is a mineralized materials that are similar to bone but does not contain any bone cells, interestingly enough, and then pulp cavity, which is the location of cells within the cytoplasmic extensions into the dentin. This is also the site where you'll find blood vessels and nerves. The tooth features from the outside is basically the crown, the neck, and the root. When we deal with the components of the teeth, you have the occlusal surface, the gingival sulcus, the cement, the periodontal ligament, the root cavity, and the apical foramen, which is the opening to the root canal. Now, there are different types of teeth: incisors, canines, premolars, molars. And when we're first born, about 6 months, everybody says, oh, they're showing their first teeth, etc. But these are referred to as baby teeth or deciduous teeth. There's about 20 of them. They're also referred to as milk teeth. Usually the teeth are all complete and in by the time you're 2 years old. But permanent teeth, you have a much higher number, 32. And these replace the deciduous teeth and start with replacement from about 6 years, and usually this ends somewhere between 17 and 21 years of age. As replacement of the baby teeth by the permanent occurs, the periodontal ligaments and the roots erode, and the permanent teeth push out the baby teeth. Now, here we go. Here's the occlusal surface, the surface where you actually meet up and crunch that jawbreaker candy or whatever. This is the enamel. This is the dentin. This would be the area we call the crown, this short area where it comes into loose contact with the gum, the neck. And then we have just below the gum surface, all the way to the end, is the root. You have the apical foramen here, the root canal, the periodontal ligament, which is really what we talked about in AP1, the joints. This is a gymphosis joint. Then we have, of course, the cement, and then we have the gingival sulcus right here. Now, you want to free up a tooth? It's not just a matter of pulling it out. It's a matter of freeing by cutting through the periodontal ligament all the way through here and here to properly extract the tooth. When we look at adult teeth, we have incisors, 2 of them, 1 canine, 2 premolars or bicuspids. This is on just one side of the jaw. And then 1, 2, 3 molars. As you can see here from this skeleton, the mandible is exposed to show developing permanent teeth here. You have erupted deciduous teeth here. You have the maxilla showing the development of permanent teeth here and here. And this is an uninterrupted permanent teeth exposed in the skull of a child. Okay. Keep in mind that you start seeing the development of these teeth. In this case, notice that the jaw seems shorter and smaller. That's because this is an entire pattern that you'd see with baby teeth. This is what you see with adult teeth, okay? Notice that you've got the incisors, lateral incisor, canine, first, second premolar, first, second, third. The third one is referred to as wisdom teeth, and in some cases can come out crooked, and thereby they have to be extracted. Now, the muscular walls of the pharynx and esophagus play a key role in swallowing. The pharynx, the throat, is an anatomical space and common passageway for food, liquid, and air. Oh, okay. Food passes oral pharynx to the esophagus. The pharynx, which of course consists of 3 parts, the nasopharynx, oral pharynx, and laryngopharynx. The esophagus is a hollow muscular tube. It's actively moving food and liquids to the stomach. Now, the mucosa of this consists of non-keratinized stratified squamous epithelium. Keep in mind, it is not to be keratinized, which would make it much more rigid. It has to be stratified because you're going to have objects move against it or side of it, having sort of an erodive effect, and that's food, etc. Similar to the cheeks. You can scrape a few layers off from a cheek, and what happens? It's replaced by the underlying cells that are the squamous epithelium. So, then you also have the muscularis mucosa, which is the irregular layer of smooth muscle. And then you have the submucosa. With the muscular layer, it's interesting to note in the esophagus that the first 1/3 the length is pretty much skeletal muscle fibers, with the second part of this esophagus being 1/3, being basically half and half. Skeletal and smooth muscle fibers, but the last third of the esophagus consists only of smooth muscle fibers. Now, the adventitia is the connective tissue layer outside of the muscle layer. Swallowing, another term for it is deglutition, and that's a complex process that starts voluntarily but progresses to an automatic process. We have three phases that make this up. The buccal, which is the bolus is compressed, and the tongue forces it to the oral pharynx. This is all a voluntary process. Once the bolus of food is in the oral pharynx, the process is all basically reflexes. The pharyngeal, the bolus forces through the pharynx past the closed glottis. Remember, the glottis is protecting so that food or liquids do not go into the larynx and into the trachea, lungs, etc. And then we have being pushed into the esophagus. At the esophageal, this is where swallowing begins, as the bolus of food forces through the entrance of the esophagus. The bolus is pushed by peristalsis to the stomach. Excuse me. You can see here's the internal nares. This is the naropharynx, oral pharynx, laryngopharynx. Notice the esophagus is back here. Here's our larynx. Here's the epiglottis, which would close over. The uvula, and this is the oral cavity. When we look at the esophagus, we have different layers. The mucosa, the layer that faces the lumen, is consisting of non-keratinized stratified squamous epithelium. Underneath that is the lamina propria. Underneath that is the muscularis mucosa. The buccal phase. Here you have hard palate, soft palate, bolus of food. It's starting to move in here. Notice that you've got the epiglottis right here. So you're in the area of the oral pharynx. Then you have the pharyngeal. The bolus is moving down across the oral pharynx, moving into the laryngopharynx. The epiglottis folds over to prevent food from going down into the larynx. And then you have the esophageal phase, which is basically now everything's on peristalsis, pushing the bolus of food down the esophagus until you get to the lower esophageal sphincter, popping it open and emptying the product into the stomach. Excuse me. The stomach and most of the intestinal tract are suspended by mesenteries and covered by the peritoneum. We've talked about that. The peritoneal cavity, the serous membrane that lines the abdominal cavity, covering most of the organs, is basically, you have a lot of potential space between the parietal and the visceral layers of the peritoneum. The visceral peritoneum covers the organs, and of course the parietal peritoneum covers the abdominal walls. We have several different structures that we've got to be mindful for. The greater omentum, which is the dorsal mesentery of the stomach. The lesser omentum, which is the remnant of the ventral mesentery between stomach and liver. And the falciform ligament. This is the remnant of the ventral mesentery between the liver and the interior wall of the peritoneal cavity. Finally, we have several other structures. The mesentery proper, which is the dorsal mesentery of the small intestine. The mesocolon, which is the dorsal mesentery of the large intestine. So, mesentery proper, small intestine. Mesocolon, large intestine. The mesocolon also fuses with the dorsal body wall as well. So you can see that in another diagram that I showed you earlier. Now, there's an interesting concept here called ascites. This is the accumulation of fluid in the peritoneal cavity, causing abdominal swelling. The risk factors include heavy or frequent, in other words, what I mean by risk, by the way, is what can contribute to possibly this onset. Heavy, frequent alcohol use, select liver damage due to medication, congestive heart failure, kidney damage. Here you see the normal situation here, and you see all of this with the free It's not really air, but basically it's a lot of open space. But unfortunately, what happens, especially when you start having liver failure, is the buildup of fluid here in the peritoneal cavity. That's ascites. And it has to be dealt with, otherwise it will impede the normal functioning of the organs in the abdominal cavity. When we talk about the formation of the digestive tract, we have to be mindful of the embryological design. You have, of course, if you look at this from 4 weeks and even look in just a space of 5 weeks, a week later, you start off with, of course, the spinal cord, you have the dorsal mesentery, you have the digestive tract, and then you have the ventral mesentery. What you'll note, if you go a little bit further, is you have the parietal peritoneal, so we're going back all the way to the beginning in the embryological development. And then you have, of course, the visceral peritoneum here and one here, holding the developing liver right there. And you can see how some of this gets shifted as we go further into development. Here, of course, is pancreas, stomach, greater omentum, lesser omentum. If you take a look over here, we have one of the ligaments that comes off from the liver and attaches to the body, and that's the falciform ligament. The lesser omentum here, which is a connection ligament basically between liver and the lesser curvature of the stomach. The greater omentum attaches to the greater curvature of the stomach. We have the small intestine and the mesentery proper, the large intestine, and the mesocolon. So, if you see this as if someone opened up the abdominal cavity, nothing was removed, and minimal amounts of cutting, as you can see, here's a greater omentum cut. What do you see? You see all of this tissue around, holding all of these organs that basically are kind of loosely defined in the abdominal cavity. They move around a little, but not to the point where their movement would be injurious or interfering in normal function of the small or the large intestine, etc. Excuse me. Here is a side view, which I think is very helpful. If you notice, here's the stomach. Here is the greater omentum, which sprawls down, loops back up, and attaches itself to the transverse colon. Here's the lesser omentum, connected to the stomach, then to the liver. So a lot of these organs are almost as if they were floating out in the abdominal space. They have to have some anchoring. Usually it's to the dorsal wall. Few cases it is the superior wall, but for the most part, there are attachments upon attachments upon attachments to some structure here. Sorry about that. Now, the stomach is a muscular, expandable, J-shaped organ with 3 layers of muscles. The stomach processes food with enzymes and acid. Chyme is the viscous, acidic, soupy-like mixture of food. In other words, when that's ready to leave the stomach, the bolus of food, no matter what you had, hamburger, hot dog, salad, has been drenched with low pH acid, has been exposed to proteolytic enzymes, and has also been beforehand ground up somewhat by the teeth. So it doesn't look exactly like the food you just consumed. But this mixture still has to go through further processing in the small intestine. In the stomach, we have several of the structures to be mindful of. The lesser curvature, which is referred to also as the medial structure. The greater curvature, which is lateral and inferior surfaces attached to the mesentery of the greater omentum. The regions include the fundus, cardia, body, and pyloric part. In the pyloric part, there is a structure called the pyloric antrum, which is connected to the body of the stomach. The pyloric canal empties food product into the duodenum, whereas the pyloric orifice is actually the stomach outlet itself. This pyloric sphincter is the muscular valve that regulates the release of chyme into the duodenum. It's not just we filled it up, made a batch of stuff, dump it all out. It goes in squirts. Okay? The rugae are the temporary folds that you're going to see in the mucosal lining, which allows gastric lumen to expand. Excuse me. The muscular layers are all smooth muscle. They include the longitudinal, circular, and the oblique. Here on the outside, you can see, the lesser omentum, the lesser curvature, the greater curvature, obviously the greater omentum. Next was the duodenum. Right here would be the pyloric antrum. Of course, you've got the esophagus attaching here. And this area that it meets to is the cardia of the stomach, the fundus, the body. Okay? When we look at this from a cadaver image, you notice that the stomach has been greatly deflated, obviously. Here we have the lesser curvature, the gallbladder, the bile duct, the pyloric sphincter is right here. The pyloric part, which probably means the pyloric antrum, okay? And you've got, of course, fundus, cardia, okay? And let's move forward here. Now, here's where you get a good representation of two things, the pyloric part and the muscular layers. If you look at the outermost, of course, you're going to have the serosa or the adventitia. But you get down into the muscular layers, you have on the outer most, you're going to have the longitudinal, middle circular, innermost oblique. Okay? This is all considered the body of the stomach. This is the fundus of the stomach. And this around here is considered the cardia. The pyloric part, when we talk about pyloric antrum, that is a term. Antrum is a term for like hallway. So we have the antrum, we have the canal, and then we have the orifice, which is controlled by the sphincter. Because what will happen is, as you have this buildup of chyme here, it's going to come out not in one big dumping, but just squirts. Squirt. Okay, we can handle that. You don't want to have it fully empty all of its content immediately because the body wouldn't have the capability to handle that incredible amount of acidic chyme. Now the rugae, as you can see here, these are the folds. So think of it this way, just like you can see some of those collapsible balloons. When you start filling it up, all the wrinkles and ripples, etc., start flattening out and the balloon expands. Now, the stomach receives food and liquids from the esophagus, aids in mechanical chemical digestion. We've talked about that. 4 major functions of the stomach: temporary storage of ingested food, mechanical digestion of ingested food, chemical digestion of food via enzymes and acid, production of intrinsic factor. Intrinsic factor is extremely important. It's a glycoprotein that's involved with grabbing onto and sequestering vitamin B12 so that it can be properly absorbed in the body. Okay? Layers of the stomach wall, as I mentioned to you before. Mucosa, submucosa, the 3 layers of the muscular layer, and then the serosa. Gastric glands, these are located in the body and fundus of the stomach. Gastric pits, the openings of the glands to the lumen. The parietal cells secrete intrinsic factor. That, as I said before, is a glycoprotein necessary for the absorption of vitamin B12. And of course, they also secrete hydrochloric acid. The chief cells, these secrete pepsinogen. This is the inactive enzyme. The acid, HCl, which is hydrochloric acid, converts pepsinogen to pepsin. That's the active proteolytic enzyme. Also in infants, it's interesting to note that the stomach will be the site of production of renin and gastric lipase, which assist in milk production. G cells are also another name for the enteroendocrine cells. The parietal cell synthesis of hydrochloric acid keeps the stomach at a pH of about 1.5 to 2.0. The process of acid synthesis is ions of hydrogen and chloride are transported outside of the cell and combined to become hydrochloric acid. Now, as we've been dealing with this, one or two points here that come up. If you recall, we've talked about this in prior classes, carbonic acid. And carbonic acid can be broken down to carbon dioxide and water, or carbonic acid can also, in a reversible manner, also break down to bicarbonate ions and hydrogen ions. Carbonic acid breakdown provides hydrogen ions and bicarbonate ions for this situation. The bicarbonate ions are expelled into the blood and account for the alkaline tide in the stomach blood flow. The alkaline tide flux is compensated by the pancreatic acid tide, and this is due to the pancreatic synthesis of bicarbonate ion solution from the acinase cells. Here we have the lining of the stomach. Now, a couple of things. You'll notice the layers, serosa, longitudinal muscle, circular muscle, and these spurts here at a different angle are called the oblique layer. You have here the submucosa with, of course, the lymphatic vessel, artery, vein. And then you have these structures here. The simple columnar epithelium, they have a lamina propria and then a muscular mucosa. Okay? And in these little crypts or in these little gaps and grooves in here are cells that will make the various substances. Pepsinogen is made as an inactive enzyme, and then it gets converted by exposure to acid to pepsin. All right, it's sort of like the one of the alchemist's dreams was to make an acid that would dissolve everything. Well, if it dissolved everything, how could you contain it? And there is a similar contradiction here. If you make an enzyme that would destroy proteins, how stable would it be inside of the cell? Wouldn't it start destroying the proteins inside the cell? Yep. So what cells do is they export out an inactive version. This is exocytosis. And then once the inactive protein's outside of the cell, it will be acted on by another agent to activate it. Hence, hydrochloric acid will activate and convert pepsinogen to pepsin. Here I have the various cells of the gastric glands. You have chief cells, G cells, and parietal cells. All of this will be mixed up as it's going up through the neck and up through the mucous epithelial cells. What happens in a parietal cell to make hydrochloric acid is pretty neat. What happens is you get carbonic anhydrase, which will take carbon dioxide and water, converted to carbonic acid. This will dissociate into bicarbonate ions and hydrogen ions. The hydrogen ions will be pumped out of the cell. The bicarbonate ions will be pumped out of the cell, but this will be going into the interstitial fluid and eventually the bloodstream. The hydrogen being pumped out of the cell into the lumen of the gastric gland is met also by a very interesting other ion, chloride. How did chloride get in here? Well, it's very interesting because you only have one ion of hydrogen and one ion of chloride gives you one molecule of hydrochloric acid. So I'm going to pump out that one molecule of bicarbonate ion. This is an antiporter here. I pump out the bicarb. I bring in and exchange another negatively charged ion, chloride. They will eventually pass through by a basically a channel protein, and when they meet together, they will form hydrochloric acid. Now, the intestinal tract is specialized to absorb nutrients. So we're going to start moving into there. Here's the key point, though. You increase the surface area to absorb nutrients. Levels of enhanced absorption include the circular folds, the plicae circularis. Then the next level of enhancement is the intestinal villi. Then the next level of enhancement on each of the villi are the microvilli, the extensions of the epithelial cells, referred to as the brush border. And also present are enzymes embedded onto the brush border to help break down nutrients. So, the layers of the small intestine start off from the lumen to mucosa, layers of the muscularis mucosa. They will actually cause the villi to move back and forth to increase exposure of the epithelial cells to the liquefied chyme. The lymphoid nodules present in the mucosa, and we have the submucosa, muscular layer, and serosa. Now, the intestinal glands, these are also referred to as intestinal crypts. These are at the base of the villi. And so in the villi structure, you're going to have these lacteals, these extensions that pass through the center of the villi. They have a central lymphatic capillary, which transports fatty acids via protein lipid packages, the choliomicrons. They'll be transported to the thoracic duct and to blood via the left subclavian vein. So what I'm saying here is this. You've got this little central capillary, it's blunt, one way. So anything that's really heavily lipid that could not get transported through the capillaries of the bloodstream can get transported by the capillaries of the lymphatic system. Capillary network, you're going to have, for blood capillary network, monosaccharides and amino acids are absorbed and transported. And they're transported via the hepatic portal system to and through the kidney, through in the liver. Here we have this rendition, which you saw earlier, of the circular plicae circularis, the villi, and the breakdown there, including the structural elements that are inside of the villi. Basically, you have in the green, of course, you're going to have a lacteal because that's lymphatic. And then, of course, red and blue, you have that little mesh up in there. That's the capillary network for the blood. And you see that right here. By the way, the yellow here would be a nerve. Okay? So keep in mind, fats get transported by the lacteal, amino acids, monosaccharides transported by the blood. Now it's interesting that you should look down here at the bottom of one of the crypts in a structure called the Paneth cells. They play a role in innate immunity, that's non-specific immunity, and they release several important materials. One of them is defensins. The second one is lysozyme. These secretions will kill some of the bacteria and help maintain a healthy microbiome. That is a microbial ecosystem. When we look at a large structural approach, the small intestine is divided into the duodenum, jejunum, ileum. The small intestine plays a key role in nutrient digestion and absorption. So the segments of the small intestine are the duodenum, which is involved, they kind of call it the mixing bowl. It receives chyme from the stomach and digestive secretions from the pancreas and liver. It's only the first 18 inches or so. Then you have the jejunum, which is where the bulk of chemical digestion and nutrient reabsorption occurs here. And has numerous circular folds, and the villi are abundant and very long. In the ileum, this is the final segment of the small intestine, the villi are relatively stumpy, so they're shorter. The submucosa has aggregated lymphoid nodules, which protect the small intestine from large intestinal bacteria. And then we have the ileocecal valve. That's the sphincter that controls the flow of material from the ileum into the first part of the large intestine called the cecum. And you can see that here. So here we have the first part, which is the duodenum. And then we move through this very nicely. And that's the jejunum. And then we come up to what's called the subsequent side section here, or the lower part, the inferior part on some of the diagrams, and that is the ileum. The ileum will empty into the cecum, the first part of the large intestine. And you can see some structural and morphological differences between each one of them. You see the duodenum, it has more duodenal glands. It does have some circular folds. Jejunum is rich with circular folds. And also you'll note when we get to the ileum, the presence of aggregated lymph nodes. How's that? Good. Remember, as in all cases, if you have questions later on, when we have our weekly meeting, please bring them. Now, several hormones regulate digestion, and I encourage you to review the flow chart and know all the hormones and their roles. The key ones here are gastrin, secretin, gastric inhibitory peptide, cholecystokinin, CCK, vasoactive intestinal peptide, VIP, and enterocrinin. Now, each hormone helps to synchronize the digestive activities and maintain protection from various secretions. So think about it this way. Once the food enters the stomach, you've got to expose it to acid, expose it to digestive enzymes. As it's broken up sufficiently, the liquidy part gets to be expelled out of the stomach into the duodenum. And what happens there in the duodenum, you have to coordinate secretion and release of bile, secretion and release from the pancreas of bicarbonate solution mixed with certain types of enzymes. When those enzymes and other materials are released, one, the acidic chyme gets neutralized in its low pH by the bicarbonate solution. And two, you're going to start breaking down the more complex molecules of triglycerides, polypeptides, and polysaccharides into subsequent building blocks. All of that, plus the coordinating of absorption, really is involved, and also to turn off each of these processes when they're not needed. Gastrin is secreted by G cells in the pyloric antrum and enteroendocrine cells in the duodenum. Okay? The functions include that you're going to have basically increasing stomach motility and stimulating gastric acid and enzyme production. Secretin, released when chyme arrives into the duodenum. Primary effect is that there will be an increase in the secretion of buffers by the pancreas, which increase the pH of the chyme. We want neutralization of that very deep acidity. GIP, gastric inhibitory peptide, that's secreted when fats and carbohydrates, especially glucose, enter the small intestine. And this is inhibitory of gastric activity, and it's accompanied by the stimulation of insulin release at the pancreatic islets. Cholecystokinin, CCK, is secreted when the chyme arrives in the duodenum, especially when the chyme contains lipids and partially digested proteins. What happens is that CCK in the pancreas accelerates the production and release of all types of digestive enzymes, also causes a relaxation of the hepatopancreatic sphincter and the contraction of the gallbladder, resulting in the ejection of bile and pancreatic juice into the duodenum. Vasoactive intestinal polypeptide or intestinal peptide, VIP, stimulates the secretion of the intestinal glands, dilates regional capillaries, and inhibits acid production in the stomach. And finally, enterokinin, which was released by the enteroendocrine cells of the duodenum in response to stimulation by the vagus nerve center before chyme reaches the pyloric sphincter, and also when the chyme enters the duodenum. Basically, it's going to stimulate the production of alkaline mucus by the duodenal submucosal cells. This is going to act as a protection for the intestine from the acidic chyme. And you can see this entire schematic of what hormone, what is their action, and in what structure. I encourage you to take time to review it. Now we talk about central and local mechanisms that coordinate gastric and intestinal activities. There are three phases of gastric secretion: cephalic, gastric, and we'll get to the other one in a second, but the cephalic begins with the CNS. Think of cephalic head, okay? And it basically is preparing us for food. The CNS acts via the vagus nerve, that's cranial nerve number 10, and also postganglionic fibers, which will activate the mucus cells, the chief cells, and the parietal cells. Gastrin also gets triggered via G cell activation. The gastric phase, this is when you have food entering the stomach and enhancing the cephalic phase responses. The stimuli includes distension, in other words, basically the filling of the stomach, okay? The stretching of the stomach as food is now entering. An increase in the pH of the gastric contents, dilution during the mixing, and the presence of undigested food, peptides, and proteins. This may continue for 3 to 4 hours as acid and enzymes act on the undigested food. The local responses are included. This includes distension of the gastric wall. This is going to stimulate the release of histamine. This is going to be coming from the lamina propria, but it binds to receptors on the parietal cells to stimulate acid secretion. Now, just here's a good point. I put down Zantac. This is what happened as scientists understood the different types of histamine receptors. They found one grouping that was unique in the stomach. And by setting up a blocker, a drug that blocks that particular unique enzyme, that unique receptor, you now had the capability of shutting down acid secretion and making it a treatment for patients, and that's what happened. Now, neural responses, these are responses of stretch receptors, chemoreceptors. Many of these are going to trigger short reflexes, and those will eventually end up triggering more secretion. For example, gastrin stimulates gastric motility, the mixing, as well as stimulates gastric secretion. Then we have finally, of course, the intestinal phase. That starts when chyme enters the duodenum. The function to control acidic chyme, emptying to maintain the proper secretory, digestive, and absorptive functions of the small intestine. Now, the enterogastric reflex, this is acidic chyme entry into the duodenum, triggers the stretch receptors and chemoreceptors to inhibit gastrin production. Gastric contractions play a role in the contraction of the pyloric sphincter. Arrival of lipids and carbohydrates. Now, if this is getting a little dry, think about it this way. Different components, what they call macromolecules, or macronutrients of food, are going to trigger certain neural signals to make sure that you have the proper enzymes and, for example, also emulsifying agents to properly break down these major components of food. So proteins, lipids, carbohydrates. Once you have lipids and carbohydrates arriving, they will stimulate the secretion of CCK and GIP. You should be getting a decrease in pH below 4.5 that stimulates the secretion of secretin. And partly digested proteins in the duodenum stimulate gastrin secretion to speed gastric processing. The rate of chyme movement into the small intestine is fastest when the stomach is greatly distended and the meal is low in protein. Okay? Hence, large meals with low protein, large amounts of carbohydrates, alcohol, or caffeine leave the stomach quickly. And also, alcohol and caffeine stimulate gastric secretion and motility. So, you can see this chart here, and you think about it. The central nervous system, sight, smell, taste of food, thoughts of food, boom. There's the cephalic phase. Gastric phase. Local responses on this side. Neural responses here. All of this is going to act in a process to enhance neural stimulation and hormonal stimulation. And finally, the intestinal phase. Now, think about this. We've already started processing the food, starting to let it get into the intestine. What's so critical? The types of nutrients and how they will trigger certain specific secretions of certain hormones. The key point: break down the material as quickly as possible. Reverse the low pH of the solution and basically begin the uptake of the simple molecules that are nutrients, amino acids, glucose, for example, fatty acids, things like that. Okay? Now, the central gastric reflexes are triggered by stimulation of stretch receptors in the stomach. Okay, purpose is to accelerate small intestine movement of contents. If I bring in more food, does this make sense? If I bring in more food, I've got to move whatever is in the gastrointestinal tract forward also. So, one, we have the gastroenteric reflex. Stimulates motility and the secretion along the length of the small intestine. Then we have the gastroileal reflex. This triggers the opening of the ileocecal valve and transfer of contents from the small intestine to the large intestine. So what am I saying? Come on, push this stuff down the assembly line and let's keep going. The central gastric reflex we can see here, and we have in it basically the gastroileal reflex triggers the opening of the ileocecal valve, allows materials to pass from small intestine to large. And let's not forget also, the ileocecal valve regulates that passage materials into the large intestine. Now, the large intestine stores and concentrates fecal material. What are its functions? Well, reabsorb water. I talked about this before. You have to take the fecal material, remove or recover a lot of the water that was used in the processing of it. You will compact the intestinal contents into feces. You absorb important vitamins generated by bacterial action. There are some bacteria that develop and make certain B vitamins. Storing the fecal contamination prior to defecation. The large intestine is going to consist of a cecum, which is the entry into the large intestine, begins the compaction process, and the appendix, which is the vermiform appendix. The mucosa and the submucosa are dominated by lymph nodules. By the way, those lymph nodules would be referred to as MALTs, okay? So mucoso-assisted lymphatic lymphatic tissues, excuse me, MALT. Now in the colon, a couple of different factors from when you compare that to the small intestine. The colon's larger diameter, but has a thinner wall than the small intestine. It consists of an ascending colon, a right colic or hepatic flexure, a transverse colon, left colic, otherwise known as splenic flexure, the descending colon, the sigmoid flexure, and the sigmoid colon. Now, the rectum is a storage of fecal material, and that's the movement into the rectum triggers the urge to defecate. There are some features to be aware of. The omental appendices, these are teardrop-shaped fat deposits attached to the serosa. The teninae coli, these are 3 longitudinal bands of smooth muscle that are deep to the serosa. We have also the haustra, which is a series of pouches in the wall of the colon and allows for expansion and elongation of the colon. We've also mass movements. Now, what mass movements are a result of the peristaltic contractions. They begin at the transverse colon and push materials along the distal part of the large intestine. One other thing to note, walls of the large intestine lack villi. You get a little bit of absorption besides water. There are a few ions, mostly lesser importance, but not a whole heck of a lot else goes through absorption here. You do want to note from your previous studies in veins and vessels, notice that you have here the superior mesenteric artery, which comes down, sweeps up, and you have also the inferior mesenteric artery, which comes down here and sweeps across as it covers the blood vessels, the arteries. And then when you start leaving, you're going to want to take that water away. So you're going to have a blood vessel supply. And that should help you there. A couple of other unique structures, okay? Here is that ileocecal valve. Here's that vermiform appendix. Now, they used to always think that the appendix was useless. The appendix now is considered to have several functions. One, it has a lot of Peyer's patches in it. So it's involved with maintaining some of the immunity and monitoring for diseases and pathogens in the digestive system. Two, and one theory came up with this. It is a storage site to reboot your microbiological ecosystem in your gut, which is a fancy way of saying this. Let's say you had severe diarrhea and you flushed out a lot of the microbes that are essential for the function of your gastrointestinal system. No frets, because the appendix has a small collection of all of them stored in itself, and it will release them when it's deemed important. Okay? Notice that you're going to have the other significant parts of the colon. Ascending, the right colic flexure. Now, by the way, the right colic flexure, the name hepatic flexure, the liver is over here. The transverse colon, the descending colon, the sigmoidal, so think of sigmoid as S. And then you have the rectum right here. And that's sort of the storage site. A couple of other little points here. Notice that there are these very significant but very small and weak, relatively speaking, bands of smooth muscle called the tendae coli. You have also these outpocketings of the walls called haustra. You also have these little fat tabs called alimentary appendices. So let's continue here. As I said, large intestine compacts fecal material. The defecation reflex coordinates the elimination of them, of the feces. Also, you're going to find that the large intestine has lots of goblet cells for mucus secretion, which acts as a lubricant for fecal material. The rectum, well, we have to look at this. We have the proximal rectum, which is consisting of columnar epithelium and consists of the anal canal. The anal columns are longitudinal folds. These are margins that contain stratified squamous epithelium. The internal anal sphincter is composed of smooth muscle, not under voluntary control. The external anal sphincter, composed of a ring of skeletal muscle under the voluntary ring. This is how you get kids to recognize, quote, they've got to learn how to do potty, because the inner anal sphincter is all smooth muscle. That's not under voluntary control. It's all automatic. The external is controlled under voluntary circumstances. We learn how to do that, and then we learn how to empty our bowels. Now, the anus is the exit of the anal canal and contains keratinized, stratified squamous epithelium. Now, a couple of other things that occur in the large intestine, just to keep you aware of this. You have water absorption, yes. Bacteria also will produce select vitamins in there, and they will be absorbed. Also, you will have the production of various bacterial products, including the gases ammonia, scatol, indole, and hydrogen sulfide. When we talk about the defecation reflex, this is an involuntary response to stimuli in the large bowel, the rectum, and that results in a bowel movement. This requires two reflexes in a positive feedback loop. The short reflex, this is triggered by fecal distension and stimulation of stretch receptors in the rectum that trigger peristaltic contractions that move fecal material into the anus. The long reflex is a spinal reflex coordinated by sacral parasympathetic motor nerves. This reflex stimulates mass movements to push feces toward the rectum from descending colon and sigmoid colon. The final steps really are to force the elimination, require the Valsalva maneuver. This is a procedure, the Valsalva maneuver, to increase the intra-abdominal pressure by tensing intra-abdominal muscles, while at the same time closing the glottis. So it's as if someone says, take a deep breath and bear down. Now, I bring this up because in certain disorders, individuals do not have the capability for the Valsalva maneuver. And this is important because without building up that intra-abdominal pressure, it's going to be very difficult for them to have a bowel movement necessary for their elimination of the waste material. Here again is the digestive system, and this is in the large intestine. Notice that you have simple columnar epithelium, you have intestinal glands, goblet cells, the muscularis mucosa, the submucosa. Notice that you have the aggregated lymphoid nodules. And then down here, you've got only just a little bit of the muscular layer. You have a circular layer here, all smooth muscle, by the way. But then you have the longitudinal layer, which really consists of the tennae coli. And notice that it's only in a couple of patches. It's just one real complete patch right here. Okay. Here is the anal canals, the anal columns, the internal anal sphincter, smooth muscle, the external, and that's skeletal muscle. And here are a couple of the vitamins. Yes, you need to know them. What you have to think about is this, that you have a variety of microorganisms. This is what we call the microbiota, or the microbiome. And it's interesting. These organisms live without causing disease in your gut. Okay. What they do is they take some of the undigested products and they use them for fuel or whatever. They will probably make some of the materials like vitamin K, vitamin B5, and biotin. Now, what you'll also notice is organic waste such as urobilinogen and various toxins generated by the bacteria will remain in the colon. Okay? You have over 1 liter of water is reabsorbed through osmosis. So you started with 1,500 mils of material and you end up with ejecting only 200 milliliters of feces. Okay. One of the other things to keep in mind is that in the intestine, if the intestine is breached, what I mean by that is if there's a puncture in the large intestine and some of the contents get into the peritoneal area, this can be a serious situation that can lead to death. Case in point in history, 1981, when Pope John Paul in Rome was shot by the Turkish assassin. What happened there was the bullet went through the large intestine. It perforated the large intestine, causing contamination of the abdominal cavity. Well, keep in mind, contamination I'm talking about is, live bacteria, and suddenly they've been put into an abdominal cavity that's moist, dark, lots of nutrients, etc. So they had to treat this very seriously, and it was very difficult for a while. They had to treat him with a lot of antibiotics. They had to close up the damage. Sometimes what happens is you also have what they call adhesions, where some of the tissue starts growing from one side to the other and creates blockages. And that unfortunately happened several months afterwards. Doctors had to go back in. What the doctors did in the beginning was they had set up an ostomy bag. So it was easy to have the waste material leave. The reason they did that was to provide for John Paul II's recovery of his intestinal structure. And then they were able to close up the ostium and resection the entire bowel, the colon. And he was then afterwards relatively healthy. But when you have a puncture like that, just like when you have a diverticuli, which is a small extension on the wall of the large intestine, diverticuli is not in and of itself dangerous. The diverticuli that gets irritated and becomes inflamed, that's where you get diverticulitis. If it ruptures and bursts, it's the same thing as having a burst appendix. It's contamination. You've got to go in. You've got to remove all the material. It can cause individuals to go into shock, sepsis, really a bad situation all around. It takes a while for the person to recover. I hope that gives you some perspective there. Let's talk about the neural process here. Basically, we've got 2 reflexes for the voluntary relaxation of the process here. And the defecation process occurs via several reflexes. Basically, when you start off, you're going to have the feces move into the rectum. They cause distension, in other words, stretching, and stimulation of the stretch receptors. This information is going to get passed back up to the brain via the short and the long reflexes. The short is basically triggering a series of peristaltic contractions. All the material gets moved from the rectum and pushed toward the anus. Okay? The long reflex, this is a spinal reflex coordinated by the sacral parasympathetic motor neurons. This information goes up to the spinal cord to a degree. Some of it goes right up to the brain. You have the voluntary controlled relaxation of the external anal sphincter here to occur at a convenient time. The internal may relax somewhat, but you still have the external, which is the skeletal muscle here versus the smooth muscle here, controlling when you're going to have defecation. Now, we've been spending all this time in the tubes and we forgot all about, yes, the accessory digestive organs. Don't worry, it's not going to be that long. But we have to keep in mind something here. We're talking about exocrine glands. These are glandular structures, and their secretion is via a duct system. Okay, so they're all going to have one type of duct or tube that travels with the secreted material from the organ to another site. The accessory digestive organs include the salivary glands, gallbladder, pancreas, liver. And we can see where they're located. Here are the salivary glands, the liver, the gallbladder, and the pancreas right down here. When we talk about the digestive and metabolic functions of the liver, they are massive. I mean, not just synthesizing and secreting bile, you're talking about storing glycogen and lipids, maintaining normal concentrations of glucose, amino acids, and fatty acids in the bloodstream, synthesizing and converting nutrient types, so conversion of carbohydrates to lipids, etc., as well as synthesizing, releasing cholesterol bound to transport proteins, inactivating toxins, storing iron, and storing fat-soluble vitamins. Remember, the fat-soluble vitamins are vitamins A, D, E, and K. But there are other major functions: synthesizing plasma protein, synthesizing clotting factors, phagocytizing damaged red blood cells. Now, this is via the stellate macrophages, otherwise known as the Kupffer cells. Okay. Storing blood, absorbing and breaking down circulating hormones and immunoglobulins, as well as absorbing and inactivating lipid-soluble drugs. But let's look at the saliva first. Saliva lubricates, moistens, and protects the mouth and begins carbohydrate digestion. There are three major pairs of salivary glands that secrete into the oral cavity. Saliva really is a mixture of glandular secretions, including water, ions, amylase, antibodies, but that's the IgA type, keep that in mind, and mucin. It acts as a buffer and a lubricant for food and maintains moisture in the oral cavity. Parasympathetic stimulation enhances the secretion of the saliva. The salivary glands consist of, well, sublingual glands, which produce mucus secretion, the sublingual ducts, which open on either side of the frenulum of the tongue, the submandibular glands, and these submandibular glands are cells that secrete buffers, mucin, and salivary amylase. Submandibular glands have a duct. This opens on either side of the anterior margin of the frenulum of the tongue. Then we have parotid glands, and these produce serous secretions with lots of salivary amylase. When you have a microscopic view, you can see there's a difference in mucus versus serous cells. Mucus are going to basically produce mucin, water, and buffers. Serous, though, produce a salivary amylase and lysozyme, whereas the duct cells will produce secretion of buffers as well as some of these antibodies, the IgAs. So saliva is really important because it keeps the pH of the mouth at about 7.0. And you can see the design here. Here's the parotid gland, here's the parotid duct, here's the submandibular glands here, here's the submandibular duct, okay? Here is the sublingual gland right here, and there you have the release area, the submandibular ducts. If you take a stain on these, now this is light microscope at 6,000 magnification, 600 magnification. The duct cells are all going to be somewhat either simple cuboidal or simple columnar. Notice the lumen. The mucus cells differ from the serous cells. The serous cells, you've got basically these nice, it looks almost like simple squamous, no, excuse me, not simple squamous, simple cuboidal cells with a small lumen here. But here you notice the cells are right here, but there's no real deep staining. It doesn't stain well here. These are the mucus cells here and here. The only stain you might pick up is from the nuclei. Now let's get into the liver. It's the largest visceral organ. It's divided into left, right, caudate, and quadrate lobes. I do want you to review those diagrams and be familiarized with the structure. It is the largest visceral organ. It has a coronary ligament that's continuous with the falciform ligament. The falciform ligament marks the division between the left and the right lobes. We can see that we have a left, right, caudate, which lies on the posterior surface, and quadrate lobe is located between left lobe and gallbladder. The bile duct carries bile away from the gallbladder and liver. The gallbladder is really temporary storage site of bile. Here you have the liver in this image. Notice that you can see very clearly the inferior vena cava, the aorta, the porta hepaticus, okay? And you should have somewhere around here the lesser omentum right here. Now, this would be the right lobe, this would be the left lobe of the liver, and you can see this in a cadaver image as well. From the diagrams, you can see left lobe, right lobe, bare area, the coronary ligament, and the falciform ligament. This is the area over here is referred to as the round ligament, and on the side here, you can see where the gallbladder is, which should be right here. Turning this to the from the anterior surface to the visceral surface, you still see the ligaments. You still see the caudate lobe, the right lobe, the quadrate lobe, as well as the gallbladder, bile duct, the hepatic artery proper, the portal vein, and the porta hepaticus. This is important. Remember that nutrients, after they've been absorbed by the small intestine, get transferred by this superior mesentery to eventually turn into the hepatic portal vein. And this massive amount of blood with amino acids and monosaccharides gets delivered into the liver where it gets processed. Some of the nutrients and some of the amino acids get absorbed. Oh yes, and if let's say you had some material like contaminants or toxins or something in your food, it probably got transported also from the intestine to the blood to the liver to be detoxified. Okay, this accounts in part for why we have an extensive and complex blood supply. Now, the liver has lobes. They're divided by connective tissue, and they end up being in the range of about 100,000 lobules. Lobules are about 1 millimeter in diameter. They're surrounded by 6 portal triads. We have the interlobular septum, which are really separations between the adjacent lobules. The portal triads consist of the interlobular vein, interlobular artery, and interlobular bile duct. Arteries and veins deliver the nutrients via blood to the liver, sinusoids, of the adjacent liver lobules. Now, remember, sinusoids are the most porous. A lot more material goes in and out via liver sinusoidal capillaries. So the blood vessels lack a basement membrane. They resemble fenestrated capillaries, but they've got a lot more openings or potential of passage of larger molecules and unique sizes and things like that. When we talk about liver cells, we're calling them really hepatocytes. And they regulate the solute and nutrient levels and absorb or secrete molecules as large as the plasma proteins. As I mentioned to you a minute ago, the hepatic portal system. This is the venous portal system where the hepatic portal vein receives blood from the capillaries of the intestinal viscera, delivers that nutrient-laden blood to the liver sinusoids. Blood flows from the interlobular vein and interlobular artery through the liver sinusoids, so you can have absorption and secretions occurring. Sinusoidal lining includes stellate macrophages. These are referred to as Kupffer cells. Remember, these are fixed macrophages that will monitor and destroy things that are not supposed to be there, pathogens and everything else. Fixed phagocytotic cells, cytic cells, these are the monocyte macrophage type that engulf pathogens, cell debris, damaged red blood cells, as well as storing iron, some lipids, and actually some heavy metals are absorbed from the digestive tract, tin and mercury particularly. The central veins collect blood to be sent to the inferior vena cava. Now, hepatocytes secrete bile, and that's collected by the bile caniculi. Bile contains what they call acid salts. We'll get into this in a minute. Bile caniculi emerge to form the bile ductules, which carry bile to the interlobular bile ducts. Here you can see a nice diagram that gives you the portal triads. Here, 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 here. Portal triad consists of an interlobular bile duct, that's the green, interlobular vein, that's the huge blue structure, and the interlobulary artery. At the same time, you can see this central vein right here. So everything heads in this sort of direction. You also have bile going in the opposite direction, heading toward the bile duct. Okay, so basically the flow is going from these very small bile ductules and then getting following into and emptying into the interlobular bile duct. Blood, on the other hand, is leaving from the interlobular vein, going down, passing by the liver sinusoids, stellate macrophages, etc., and eventually gets to the central vein right here. Okay? This one whole section is called an interlobular septum. Now, what about the gallbladder? This stores and concentrates bile. Gallbladder structure consists of the fundus, body, neck. The hepatic ducts, they're right and left. They merge to form a common bile duct. Now, the cystic duct is the duct directing bile to the gallbladder. The bile duct carries bile, meets with the hepatic duct at the duodenal ampulla, which is also referred to as the chamber, and the ampulla part of the duodenal papilla. And this is where you have the release of bile and pancreatic juice, and this is controlled by the hepatopancreatic sphincter. Now, keep in mind several other things that you need to keep in mind regarding bile. Bile is an emulsifying agent. What it does is if you think about this like large blobs of grease, if you ever washed greasy dishes, you get these big blobs of grease, and you add a little detergent, bloom, and you get these smaller blobs now. That's what an emulsification agent does. It breaks down the larger blobs into very smaller blobs to enhance the lipase, which is the enzyme that breaks down these blobs of lipids, to enhance their surface area contact for lipid breakdown. Another way to think about it. Without bile, tell me more without bile. Certainly. If you had a big, fat banana split or a hot fudge sundae with lots of ice cream and lots of whipped cream, etc., and you didn't make bile, you would have passing out of your stools what is called stearorrhea, meaning fat in the feces. There's just no way that lipase would be able to break down all of those lipids, and the lipids would pass out of you, okay? The problem with bile is that it can become concentrated in the bile duct or the gallbladder, and it can precipitate into crystals and then what we call stones. See, bile is not just bile pigments. Bile contains a variety of water and ions, cholesterol, etc. Now, this mixture, if it just sits in the gallbladder for a period of time, you start losing some of the ions and water, and it starts to concentrate and concentrate until some of the more fatty and some of the more mineralized materials precipitate out and start forming these crystals or stones. And once you've got a stone, it just continues to keep building. All right? And if it blocks passage of fluid through either the bile duct or the gallbladder itself, it becomes exceedingly painful for the patient. Now, it's interesting to note that CCK, cholecystokinin, is the hormone that triggers the ejection of bile into the duodenum. CCK plays the role of contraction of the gallbladder, dilation of the hepatopancreatic sphincter, and ejection of bile through the duodenal ampulla. Okay? Here we see the gallbladder, the fundus, the body, the neck. Here we have the cystic duct. Here we have the common hepatic duct, and you have a right and a left hepatic duct. And then, of course, you got the general bile duct now. Okay? Now, as you notice, as we pass through this, and this was a cut edge of the lesser omentum, we eventually have this come underneath and around and through the pancreas. Where you get up close here, you can see the bile duct meeting up with the pancreatic duct. And they come together at this hepatopancreatic sphincter. And you have, of course, the duodenal ampulla here, the duodenal papilla here, and then, of course, the intestinal lumen right there. So basically, once you control this, these fluids come together and get discharged at the same time into the duodenum. Everybody with me so far? Now, as you move through this, one of the things you got to keep in mind is this. Okay, if I lose my gallbladder, can I still make bile? Yes. You just can't have the vast, heavy, fatty meals that you may have had before, because now your reserve of bile is now no longer there. You will have secretion of bile. You just have to keep that balanced out. Here's the liver. It makes the bile continuously as it remains in the gallbladder, becomes more concentrated, and we go through this process here. But before I go from there, I'll just give you one other quick situation. There was a dieting company in around 1992, and it's always a procedure that some dieting companies do, and they basically have a strategy for nutrition called ultra-low lipid foods. Now, these ultra-low lipid means that they've really been able to remove most of the fat, most of the lipids from the foods, and so you get these diets that are ultra-low lipid. There's a problem with them. In 1992, this particular company went bankrupt. They had to file for bankruptcy because they were killed. They were really bombarded with class action lawsuits for all the people that had used their ultra-low fat diet that ended up with, ready? Gallstones. Now, they would have it on news magazines like 2020 and all these other organizations and all these other news items. And so this company went really into shambles. The bigger kicker was a 15-year-old who had to have her gallbladder removed due to gallstones. It's kind of like, Wow. What you need to be aware of as an allied health person for the future is that ultra low lipid diets are always going to backfire. You have to have some lipids in your diet. Maybe that doesn't mean that you have to have 15 pounds of ice cream a week, but it does mean that you have to have some lipids for dietary needs, etc. And to also make sure that you're using that bile for what it's needed for, which is the breakdown of the lipids. Let's move into the pancreas. The pancreas has vital exocrine and endocrine functions. Remember, we've talked about endocrine at the beginning of the course. That is where you have the islets of Langerhans, the pancreatic islets that are going to produce insulin, glucagon, somatostatin, and pancreatic polypeptide. Okay, but that's the endocrine portion. The exocrine portion is that it will produce pancreatic juice, which consists of bicarbonate ions, as well as various digestive enzymes for breaking down proteins, lipids, carbohydrates, as well as nucleic acids, because there's a lot of nucleic acids in various foods, right? Every cell that you have has got some bit of DNA in it. It's got to be broken down. So the pancreas is located, it lies posterior to the stomach. The pancreatic acini cells will make this bicarbonate and ion solution. The pancreatic duct delivers the exocrine solutions to the duodenum. And then we have, of course, we've mentioned briefly already, the pancreatic islets. Now, the pancreatic lobules, these are divisional sections of the pancreas, and this is due to the connective tissue partitions. As you can see here are the pancreatic lobules, the tail, the body, the head of the pancreas. You can see how it rests neatly in sort of this curve of the duodenum. You will see the pathway where you have the pancreatic duct. You will have also an accessory pancreatic duct here. Okay, so this is collecting all of the pancreatic juice that consists of material made by the acini cells, here, etc. Okay? Ignore the fact that, yes, you've got the endocrine cells of the pancreatic islets here, except for the fact that, yes, they do exist. Yes, they are involved with the endocrine system. We're focusing on the digestive system and mostly with exocrine secretions. Here are some of the major pancreatic enzymes you're going to see. Pancreatic alpha amylase, that's going to break down starches. Pancreatic lipase, that's going to break down lipids. The nucleases, that's going to break down nucleic acids that appear in many of the different cells, such as RNA and DNA. And then proteolytic enzymes, and there's a whole mess of those. Now, let's talk about the disorders of the digestive system. They're diverse and relatively common. In the oral cavity, we have periodontal disease, disorders of the teeth, usually due to the accumulation of dental plaque. Gingivitis, this is the damage to gums due to the plaque damage. We have the salivary glands, a viral infection of the salivary glands known as mumps, but it has been reduced in its prevalence because of vaccines against mumps. For the esophagus, we have esophagitis, which is an inflammation of the esophagus. We also have the gastroesophageal reflux, which is a backflow of stomach acid that results in esophageal damage. Remember that you have a much simpler, mucosal layer in the esophagus. It's not set for protection against acids. And so when you have this reflux, it tends to cause damaging, including the possibility of esophageal ulceration because of the overexposure of that stomach acid. When we talk about the liver, well, we've got several different disorders. One of them is hepatitis, which is the inflammation of the liver, but it may be due to virus, alcohol abuse, infection, or drugs. Jaundice, which is the yellow coloring of skin and the sclera of the eyes due to the bilirubin buildup in the blood. This is usually an indicator of liver disease. When I talk about cirrhosis, this is a hepatitis indicator. In other words, liver damage. What happens, even though they say that the liver is one of the most prolific organs to repair itself, it has limitations. And a lot of times what happens is with the liver, if you're constantly having viral attack or using heavy amounts of alcohol, etc., or combine that alcohol with acetaminophen, you're going to end up with the hepatocytes dying and being replaced by just connective tissue. This is sort of the scarring that'll occur. In the gallbladder, we've talked about this, formation of gallstones, which is due to the buildup of crystals of biomaterials, which can block the bile release from the gallbladder or can block the bile ducts. When we talk about the stomach, we have gastritis, which is an inflammation of the stomach lining. Ulcers, the stomach can be, in the stomach, these can be caused by Helicobacter pylori, which is a bacteria. It took a long time for people to recognize that, and I mean well over 100 years. They first thought it was bacteria, then they thought it was a virus, and they thought it was this, then they thought it was that, then they thought it was stress. And many of the ulcers today are caused by an infection, by the bacteria, which get into the lining of the stomach. Now, remember, what protects the lining of the stomach from the actual acid is a thin layer of mucus. The bacteria, though, break down the mucus. How do the bacteria survive in such an acidic environment? They have urease, an enzyme that allows them to break down urea to make an ammonia ion coating, which neutralizes the effectiveness, the low pH of the stomach acids, allows them to survive, and they continue to basically break down the mucosal lining, which allows for killing of the stomach lining cells and the formation of an ulcer. In the pancreas, we have what is called pancreatitis. This is an inflammation of the pancreas, usually due to toxins, drugs, or a virus. In the small intestine, there is a condition called enteritis, which is the inflammation of the intestine. Diarrhea is watery bowel movements. For example, Giardia lamblia is a protozoal parasite that causes intestinal infections. What it basically will do is attach to the walls of the intestine to block absorption of nutrients. And if you have sufficient of them, you will end up with either diarrhea or stearrhea, fat in the feces, or worse. They have to be treated with a certain anti-protozoal medication to basically eradicate the Giardia from the intestine of the patient. In the large intestine, we have colitis, which is an inflammation of the colon. The concept of constipation is infrequent bowel movements, and that's usually due to hard, dry feces. And then we have polyps, which are localized growths of the intestinal mucosal wall. These can be both benign or cancerous. And you can see here, here's the gingivitis. You can see where the lower parts of the gums are eroded away, it's the teeth. You see the inflammation of the parotid glands here with mumps. Esophagitis is seen by endoscopy. You can see that red and inflamed area. This is a gastric ulcer, so that's damage in the tissue. Cirrhosis of the liver will lead to basically the replacement of a lot of the liver tissue with connective tissue, or in some cases where it's fatty, it will be just blobs of adipose tissue. Acute pancreatitis, you can see here where there are large bloody blebs replacing acini. The gallstones, if you notice, that's a dime there. So these are stones that actually exist. These crystallizations of this cholesterol, fatty material, and minerals, etc. So they can get quite large. This is the Giardia lamblia, which is a multi-flagellated protozoan. It's also known as beaver fever, folks. If you're a camper, you know you've got to decontaminate your drinking water. And you don't want to drink water nearby where beavers are making a dam, etc., because beavers are a natural reservoir of this particular parasite. This is a colon polyp seen by a colonoscopy. They've got a loop around it. This little mass there is going to be extricated for a biopsy. Okay, we've done well. You've completed this entire chapter. I know there was a lot, but you've done well. I want to encourage you to peruse the chapter review outline on pages 885 to 889, complete the review questions on 889 to 890, complete the review sheets on 846, 872, and 884. Our next chapter will be chapter 23, involving metabolism, nutrition, and energetics. And have a nice day.